Voir si c'est
On n'a
possible.
pas rangé. Ah non, ouais, ok. Ouais.
Okay, welcome back. And to begin the session, to begin the last talk of the afternoon about behavior, we can welcome Jemima Frame. You are a PhD student at the Swansea University in the Department of Life Sciences. And today you will give us a talk on combining accelerometry and GPS data to investigate grooming reciprocity in wild female Chacma baboons. So the stage is yours.
Yes, I'm presenting this. So grooming is often seen as the main affiliative behaviour in social primates, building, strengthening and maintaining social bonds between individuals. And this comes with a number of long-term benefits, including increased reproductive success, increased coalitionary bonds, access to resources, and an overall decrease in stress levels. But the act of giving grooming itself has often been described as altruistic behaviour, so there's no direct benefits to the groomer, and they actually incur costs in the process. So this includes a decrease in energy use, less time spent on other activities, and a temporary increase in stress levels immediately after giving grooming. So this raises the question, if grooming entails a cost to the groomer, why is it so prevalent among non-kin individuals? One possible explanation for this is the biological market theory, which posits allogrooming as a tradable commodity which can be exchanged for other services. So this includes access to food, access to infants, increased spatial proximity, and grooming returned back to them. So the biological market theory helps to provide an evolutionary framework not only for the emergence of grooming, but also for why specific grooming dyads emerge. So, for example, in species where there's a dominance hierarchy, we would expect individuals with higher dominance rank to receive more grooming because they have more services to offer in exchange. So my project looks at grooming reciprocity, so the act of giving grooming to receive grooming back. And there's two mechanisms for how this is done, partner control and partner choice. So partner control is where dyads are isolated and partners reward reciprocation and punish uncooperative behaviour. So this is often very fast. So within a dyad or within a pair, one individual will groom and straight away the other individual will groom back. And then there's partner choice, which is where partner switching is available and partners are determined based on the goods they can offer in return. So this is often over a much longer time frame. So one individual can groom, they can separate, do their own thing, and then eventually come back to each other and the other one will return the grooming. So the biological market theory favours partner choice as the main mechanism behind grooming, but despite this, most research into grooming reciprocity still only explores partner control due to methodical limitations. So these methodical limitations are mainly that the majority of behavioural data is mainly collected through observations at this time. So this means that data cannot be collected on all individuals at the same time. It can't be collected 24/7 on these individuals, and it relies on observer accuracy. So when we're looking at things like specific timings of grooming, it's likely to be a tiny bit inaccurate. In terms of reciprocity as well, focal observational data often is no more than an hour of focal observations, which leaves a very short time frame to actually look at reciprocity, and it only looks at partner control mechanisms. So my study species is Chatham baboons, which are located in South Africa. They are a highly social species. They can be found in groups of 15 to 200 individuals. And they each have a dominance they have multiple males and multiple females in the group, each with dominance hierarchies. So the male dominance hierarchy is decided through aggression, and the female hierarchy is hereditary. So my methods aim they aim to combat some of these issues with observational data. So we fitted a troop of baboons with collars, which collected GPS and accelerometer data. So it collected their location at every second time point and their triaxial movements at every second time point, which meant that we could devise what their behaviour was for every second. And using this, we were able to calculate who the grooming dyads were. So for every grooming interaction, we looked at which individuals were in the same location and therefore could be grooming, and of those individuals, which were giving grooming and which were receiving grooming at that same time point. So this gives us grooming data to a second level accuracy on a much wider timescale with no reliance on direct observation. So I had 3 questions from this. Firstly, does direct reciprocity occur? Secondly, is there a long-term time matching element to direct reciprocity? And lastly, if so, over what time frame does time matching occur? So for my first question, we found that yes, direct reciprocity did occur, which we expected. So we found that the occurrence of grooming by direct reciprocity was much higher than grooming by random chance. So to put it simply, within a dyad where there's individual A and individual B, individual A was much more likely to groom individual B if individual B had previously groomed individual A. So for our second question, is there a long-term time matching element to direct reciprocity? We found that also, yes, there was. So there was a significant positive relationship between grooming durations in a dyad. So this shows that the direct reciprocity extends to the duration of grooming. It's not simply they're reciprocating based on the act, they're actually tracking the time spent grooming. And interestingly, we found that rank and proximity significantly influenced this relationship. So if one individual had a higher rank, the relationship was weaker. And if the individual spent more time together, which can be inferred that they have a stronger social bond, the relationship was stronger. And both of these are evidence of partner choice mechanisms at play. Lastly, we want to look at over what time frame does time matching occur. So we used a reciprocity index, which is a score which measures the strength of reciprocity. So it's a score from 1 to 0, where 1 is complete reciprocity, so they're grooming each other the exact same amount of time, and 0 is no reciprocity, so only one individual is grooming the other. And so to look at over what time frame, we looked at the reciprocity index for each dyad over increasing time frames, so ranging from a daily scale to beyond. And we found that interestingly, the reciprocity index was increasing as we increased the time frame. So as we increase time frame, the strength of the reciprocity is increasing until it reaches a stabilizing point. However, to answer the question over what time frame, as you can see, so each dotted coloured line is a separate dyad, and there's quite a lot of variation between. So the overall line is not very representative of each dyad. So we decided to look at the dyad level. So again, we see the same relationship. So reciprocity increases over time before approaching an asymptote. But there is a lot of variation between each dyad, both in terms of their reciprocity index at the point it stabilizes and the optimal time frame. So for each dyad, we looked at the point of maximum change of the slope for their curve to identify their optimal time frame. So this is essentially the point where if we increase the time frame past this point, the reciprocity index won't change much. So this gives us essentially each dyad's true reciprocity index. But to answer the question over what time frame, there's not a clear answer. There is a lot of variation. And yeah, so but one thing is very clear, that the daily scale is not wide enough to capture a dyad's true reciprocity index. And this can be shown clearer through a heat map. So the one on the left, right, is their daily scores. So the reciprocity index calculated at a daily level, and the one on the left is their true reciprocity index, so the one where their optimal time frame is. And these are significantly correlated to each other, but they're also significantly different to each other. And it's important to note that many papers regard under 0.5 to not be reciprocity. They regard that as just grooming by chance. And in the daily scale, 40% of the dyads have under 0.5 as their score. So in traditional methods, a paper would regard 40% of the dyads to not be doing reciprocity, they're just grooming by chance. Whereas when you look at the actual true reciprocity index, only 14% of the dyads are under 0.5, which is actually only 4 of the dyads out of 28. So this really highlights that there can be a lot of inaccuracies based on assumptions which are incorrect purely because of the time frame we're looking at. So overall, I found that there was significant daily grooming duration relationship, which we did expect to find, but it has been difficult to find in past papers. There's lots of conflicting evidence, and this is likely because they were looking over much shorter timescales. So it shows that grooming reciprocity is not necessarily immediate, and it occurs over a larger timescale than we previously assumed. This really highlights the importance of temporal scale and behavioural studies, and it shows we may be missing key patterns because our current observational timeframes are not long enough. Thank you for listening.
Questions?
Hi, it was a great presentation. I just have a naive question, but you said that there was potentially partner choice. But what comes with diet? Like when you are looking at this diet, like it's just random individual that decided like to do this to each other, or then it's always between a female and a male, or
So actually, I should have mentioned this was only females, so it was all female pairs. And yes, the partner choice was at play, so not all of the females engaged in grooming together. So there are certain pairs where there's very little grooming data or no grooming data. So there's definitely dyads which favour each other compared to others.
and is it like one female like?
Yeah.
I have a question regarding the increase of reciprocity over time. Why do you think that happens? Because I assume you're following these individuals for a long time, so you might start when they've already been reciprocating, if that makes sense.
Yeah, definitely. So wait, let me go back. So it's not just like one. The daily scale was multiple daily scales, and then like 2 days is multiple 2 days. So it's an average score, not just like starting at day 0. Because you're right, they would have been grooming before that day as well. And yeah, I think it increases over time because it's kind of showing that they're not reciprocating just on the day, just within a day. They take time apart, they do their own thing, but they eventually will come back and reciprocate. It's just over a longer time frame.
One last question.
Thank you for the presentation. Okay. I was wondering, in this process, how the previous interaction that they have in a day can affect the reciprocity in the next dyad that they form. So because they all interact with, with different individuals. So did you observe anything like this, or in your data, can you analyze how what they do with the dyad in the day will affect what they do in the next interaction with the next individual?
I haven't looked into that. I definitely could. I imagine it wouldn't change much because each kind of dyad is separate. So if they groom another individual, it's completely separate to the grooming individual C, let's say. And I think that because it's over like multiple days, it means that maybe they won't reciprocate the other one immediately because they're tired, but they will still eventually reciprocate them. And you can see that for here, like each dyad has different reciprocity indexes. So some of them are reciprocating much more evenly than others. So,
yeah, I thought maybe if they encounter an individual which doesn't have a lot of reciprocity, maybe in the next time they meet a next individual, maybe they will have lower reciprocity towards these specific individuals.
between the same individual.
Sorry?
So you're saying if they don't reciprocate, they won't reciprocate back as much the next time?
Yeah, exactly. Yeah.
Yeah, potentially. But I think that also then has like dominance hierarchies and other factors have a play in that as well. So maybe they're offering other services in return that aren't grooming.
Cool. Thanks.
And the next person speaking will be Chuck Noster. Is it, do I pronounce it right?
Yeah, you can say Noster even if you want in French way.
Okay, so you are a PhD student. You work in the University of Vienna in the Konrad Lorenz Institute of Ecology. You will talk about, can social proximity predict audiovisual similarities in the courtship of spotted bowerbirds? Okay, so 1 second, the presentation is up. Yeah, the stage is yours.
Thank you. Okay, yeah, so just as it was said, I'm a PhD student at the University of Vienna. I'm supervised by Leonida Fusani and Cleona Quigley. And I'm working on the cost of courtship involved in the spotted bowerbird, which is an Australian species of bird that built a very interesting structure called the bower that you can see here on the left. And it's a structure that is built and decorated by the male with several objects like the green berries you can see here and the shells you can see in front of it. Also sometimes some artificial decorations like the trash you can see in front of this one. So the structure is, and even though it looks like a nest, it's only used in the context of courtship display, never as a nest. And the female will stand inside, as you'll see in the video, while the male is doing his courtship around her. Notice the lovely vocalization. So yeah, quite an aggressive courtship display. And the quality of the bower, the number of decorations, the quality of the courtship display, the dance they do around, and the vocalizations are all important for the mating success of the male. And interestingly, these males take a long time before they can become a bower owner, before they become territorial, 7 years, even more sometimes. And actually, I lied to you in the video. We just saw it was not a female standing in a bower. It was actually a young male. In these 7 years, before they become a bower owner, males are called subordinate males, and they are tolerated by all the males. They can play around in the bower, pretending to be females when they receive a courtship display. And yeah, the reason why this interaction is existing is quite still unknown, but one hypothesis, one benefits for the younger males might be that they are socially learning courtship. So all the skills they need to build the bower, but also to perform the courtship itself. So this is something we investigated in this paper that we published a couple of years ago with my collaborators. And I cleverly renamed it that way for the conference, just reversed. So where we looked exactly if social proximity between individuals can predict how they are visually and acoustically overlapping in their courtship display. So what do we know about learning in this species? We know that they are very good vocal learners. They are excellent at mimicking other species, especially in threat contexts. They are learning very likely to build this structure. This is something that has been shown. There's some local patterns in how the structure is built when you look at different areas of a single population. And we know also that in other species, there's some dialects, so some differences, geographical differences in the vocalizations they perform. So yes, we think that might be something going on like courtship is socially learned, and that's what we will look at now. So to study that, we work in a field site called Taunton National Park in Queensland. And this study has been done on 2 years of data, 2019 and 18. And there we color band the birds, so we can identify them individually from videos that we take with camera traps. And we take also some blood samples for them for some genetic analysis. So how to look if there's something, if something in the courtship is socially learned? We take many measurements of the courtship display, and we put them in a distance matrix like that, where we look at all individuals as lines and all individuals as columns, and then we will compare this matrix for the vocalizations or for the courtship movements and with other things like the geographic distance, the social distance between individuals, and we will compare it with a mental test to see if there's a correlation between those matrices. So for the acoustic overlap first, yes. We extracted the vocalizations from the videos of the camera traps you saw before. And we extracted one type of vocalizations, the most common one, and manually checked if they were all scras for up to a bit more than 35,000 vocalizations. So scra is the call, this type of call you heard during the courtship display we've done displayed. We had 26 individuals, 13 owners and 13 subordinates. And from these vocalizations, we extracted a lot of acoustic variables about frequency, duration, and this kind of things. To reduce a bit the noise of the data, we put all these variables into 5 components. We reduce them into 5 components of an LDA, which is some kind of PCA, where instead of just spreading the data, we spread it across a factor, which is in this case the individuals. And then we look at how the individuals overlap over these components of the LDA. So that gives up the overlap between individuals for the acoustic data. Then for the geographic distance between individuals, we looked at the favorite bower where they spend the most time, and we just look at the distance, like the Euclidean distance between these bowers. Then for yes, the social proximity between individuals, we look at the time an individual, a younger male, will spend watching another male displaying. And that's something we consider as being the potential learning time for this individual. So yeah, what are the main results? First, we observe a correlation between geographic and acoustic distance, which is expected when we have dialectal structure in the courtship. And then we have also a correlation between social and acoustic distance, which is quite interesting and quite new. That's something that's usually not easy to observe. And then we, of course, control for possible inheritance, and we don't have any correlation between genetic and acoustic data. So how to disentangle between geographic distance and social proximity as being, which one of the two could be the best predictor? So to look at it, we here plotted, you can see in this plot, the geographic distance and the acoustic overlap. And the correlation I was explaining before was basically all these data together, it's correlating. Then each point being one pair of individual here. Now, if we look at only the individuals that are socially interacting, so the individuals that are as red dots here, the correlation is staying. And then if we look at the non-interacting individuals only, so only the crosses, the black crosses, we are losing this correlation. So thanks to that, we can think that social proximity is a better predictor than geographic distance. So, yeah. It seems like social proximity can explain acoustic overlap between individuals, and it seems better than geographic distance. So this is quite a strong non-experimental evidence in the wild, and it has been done at the population scale, which is quite also rare as a study. So we can speak of something like a microdialect. And yes, it's an interesting use also of side-produced data because it's just the recordings from the camera traps from which we extracted the audio, and still we could do something interesting with it. So I think that's also relevant here. So as you saw in the title, the results were also about the movement, and here the results are a bit less as we expected them to be. So we see a correlation between some of the measures we take for the movement of the courtship display and the geographic distance, but we don't see that correlation when we look at social proximity between these individuals. And so, yeah, a hypothesis, what we think is that the measurements we have of the courtship display are very rough. It's just basically the different types of movement, and we look at metrics of these types of movements. So in the future, we have right now a master student working on automatic tracking of the displaying bird, and with that we will extract much more detailed information, and we'll look then again if there's some social learning going on in the movements of the courtship display. So with that, I would like to thank everyone, my collaborators on this project, everyone that helped, the founding bodies, and you for listening.
Hi, thank you. It was very cool. I have a question. Do the subordinate also inherit somehow the territory or not at all? And are the subordinates somehow related to the adult's owner?
So no, they are not related for what we could see until now. Yes, they can inherit the territory. This is something that still need to be studied properly because, as you saw, like they take quite a few years before being able to inherit something. So do they have more chance to inherit where they are subordinate or not is something we still need to check, but they do inherit territories. Yes.
Thank you.
Thank you. Very cool study system. I was wondering if the males do not reproduce until they reach that age when they can hold resources and perform the courtship, or is there any sneaker morphs?
Yes, there's some sneaky copulations. There's a short communication of Giovanni sitting next to you on that topic. Yes, actually they have sometimes, it's quite rare, but there's sneaky copulations going on from the subordinate where the bower owner is displaying around the bower, the female is inside ready to copulate. The owner doesn't notice it early enough, and there's a subordinate suddenly coming.
Great, I love it.
Can you pass the microphone in front, please?
Thank you for the talk, very interesting. I wonder if geography and social proximity, are they correlated?
So say that again,
Geography and social proximity, you say sometimes they correlate to the display or to the acoustics, but are they both correlated?
It's a very good question, and I remember checking that. Instinctively, I think I would say yes, but I don't remember. That's been a while now. So this kind of checks I've done, but I don't remember what was the result for that, sorry.
Okay, thanks.
Probably in the paper.
Okay, thanks again for this amazing talk. Okay, so the next person we will welcome is Elena de Miguel Martinez. You will talk about individual and broad variation in begging calls and offspring's recognition in pied flycatcher. That's it. And you are a master student in the Stockholm University and in the Whitcroft Lab. Yeah. Is it Whitcroft? Yeah. Okay. So, 1 second, your presentation should be okay. Right? The stage is yours.
Well, good afternoon. As they say, I'm going to be talking about variation in begging calls and offspring recognition. First, a brief introduction to the topic. Parental care is defined as the provision for offspring that increases offspring survival and overall fitness, and it's quite widespread across the animal kingdom. It is beneficial for parents, but it also requires a big investment of resources, so it's quite costly, and therefore parents should only provide this care to related individuals. Some cases in which care can be misdirected are when there's brood parasitism, extra pair paternity, or when offspring of different parents share the same spaces and intermingle with each other. So a way to tackle this potential care misdirection is by identifying the offspring. Given the cost, parents are likely to present sensitivity to different cues from their offspring. In birds, for example, nestlings produce begging calls to solicit care, both when they're in the nest, but also after they've left it. And sometimes there's variation in these begging calls, and this could be exploited by parents to identify their actual offspring. This has actually been shown in colonial breeding species like razorbills, zebra finches, and cliff swallows, where parents can recognize their own offspring from others by their begging calls. But in non-colonial breeding species, the risk is lower, and therefore it has been quite overlooked. However, the risk of care misdirection increases when nestlings have fledged, and how parents tackle this issue is still unknown. So this is the context in which I performed my study. We looked at how parents of pied flycatcher identify their offspring. And we state two different hypotheses that begging calls vary across individuals and nests in a way that aligns with a brood and an individual signature, and that parents can recognize their own offspring from other nestlings based on such variation. So my study species is the pied flycatcher. It's a territorial songbird that migrates to Europe to breed and that nests in tree holes and nest boxes. They present adult sexual dimorphism and biparental care. This care is given both while nestlings are in the nest and after they fledge, which happens around 14 to 16 days old. And my population was a population that was breeding in nest boxes around this research station in Sweden. So to assess the first hypothesis of whether calls vary, I performed acoustic analysis in the begging calls of nestlings that were 13 days old, which is right before fledgling. I used 10 calls per individual and extracted 16 acoustic parameters, which I used to assess whether calls vary and whether they were assignable to the nest and individual of origin. But the results showed that there's in fact variation at nest and individual level, and that calls were highly assignable to the true nest and individual. Here there's a spectrogram, which is the representation of frequency versus time. And each row represents calls from 3 different individuals of 3 different broods. And the variation between individuals is higher than the one within the same individual. So there is variation in the structure of begging calls, and they can be assigned due to such variation. But can parents actually use this to identify their offspring? To assess that, I performed playback experiments where I assessed parental response to nestlings begging calls when they were 13 days old. I presented 2 different calls, ones from their own offspring and ones from other unrelated offspring. I simultaneously broadcasted these calls for 10 minutes and assessed parental response through the distance to the speaker, and more specifically through the time that they spend closer to one speaker than the other one. So if parents were able to recognize their offspring, the results would show that they spend more time closer to the speaker that broadcasted the calls from their own individuals, which was what we in fact saw. So there is variation across in begging calls across nested individuals, and parents respond more strongly to their own offspring's begging calls. So both statements indicate the presence of offspring recognition in this species. This is the first evidence of such signature and offspring recognition in the pied flycatcher, and one of the first times that it's been shown in a territorial breeding species. A couple of things I would like to discuss is that there could be additional sources of variation, like, for example, nestling hunger level. But it's been shown that even with hunger affecting the structure of begging calls, individuality can still be detected. How and when do these signature and the recognition arise is still unknown. In order to investigate that, there would be needed cross-fostering experiments and analysis and experiments at different nestling ages to determine when do they arise. And we observe a slight tendency of females responding more to their own nestlings calls than males, but it was not significant. So further research should be required or would be required to address this issue. So the presence of this trait in these species could have good implications for other territorial species, given that this trait could be more common than we know, and it would be a good starting point to address this issue. Thank you for listening.
Hi, it's just a curiosity about your method, because you said that you simultaneously put in the playback experiment, you simultaneously put the two calls.
Yeah.
I was wondering why you chose that instead of putting either one and the other and see how often they come to the speaker.
Just because, I mean, this has been done in other species, and we thought that that would show parents it would be an simultaneous stimuli instead of biasing, like maybe they could go to one first and then the other one. So they could choose over like the two stimuli whether they want to go to one or the other one. So, but I think that could be a good possibility, and then to just change which one starts first and every time.
Yeah, no, because I could imagine that if you put only one, maybe when you put the strangers, they might not come at all, or they might still come, but maybe to a lower rate. So I think that will also be interesting somehow.
Yeah, I mean, so they sometimes went to the stranger ones, sometimes to check it out, sometimes from further away. It's just that they would go more often and from closer to the ones that were their own. So.
Thanks.
Hi. I didn't understand, I think, how you differentiated the songs. Like, what song phenotypes did you look at? Like, amount of peaks they had per second, or like frequency? What was the different thing between the begging calls in different nests?
So I look at 16 different parameters, and I combine them with a PCA to make it easier for the analysis. But I looked at frequency parameters, frequency modulation, length, bandwidth, and a couple things more. Just it's a bit long to explain everything.
And they all differ, or some of them?
No, just some of them. So for the ones that were given the most variation, or the that were driving the assignability more, were the frequency and the frequency modulation parameters.
Okay, I see. And do you know if these parameters are also used for mate recognition or perhaps for partner choice? What I'm really wondering is, this is great, but it sounds like a lot of energy again for the parents. So I wonder if this evolved for the sake of recognizing the begging chicks, or is this a byproduct of the evolution that already made these birds' brain, of like the acoustic region of the brain, really complex and amazing because of sexual selection?
Yeah. So begging calls are only used, or as far as we know, in a context of when they're nestlings and fledglings, and they're not so used when they're adults. In some species, they use begging calls in the nest, for example, for tutoring the nestlings. So the adults actually produce begging calls, but I haven't seen that they are used in a mating context. And no one has ever compared like the begging call structure with the song structure of the same individual that I know of.
Maybe we can talk later in the break. Thank you.
Thanks.
Thank you for the talk. About the result, I'm not sure I completely got what explained variation in the call. So is the nest more important to explain the variation in the code, or actually the individual component?
It's more important the nest level. Most of the variation was driven by the nest, like between 30 and 20%, and then individual level. And actually the recognition is based on nest level. We didn't test if they would differentiate individual A or individual B from their own brood.
Okay. What is your gut feeling about this? Do you think for the parents it should be more appropriate to recognize the call of their full nest or specific individuals? Would that affect their response?
I would think the whole nest, because for them it's too cognitively demanding to know which one is which, and they might not get a really benefit from this. They don't have brood division, that is when you favor some individuals over others within your brood. And they most likely use this after all the nestlings have fledged the nest, and then just to locate them or feed them. So they would need more to know that this one is theirs and not from the other nest than that they are individual A or B from their own nest.
There is not like some kind of a crossover experiment that has been done to try to see like if parents respond different to their own chicks or cross-foster chicks?
Not yet, but that's kind of like the next step.
Yeah, sounds cool. Nice, thanks.
The time for other questions. Well, if we have no more questions,
Thank you.
Okay, so we have one last talk before our coffee break. This is also the last talk of the behavioral session, and it will be given by Zoe Arrigoni. You are a master student. You are from the University of Savoie-Mont-Blanc in the INRAE laboratory, and you will talk about the behavioral impact of road dust and tire wear particles on the grammaried and potential functional consequences of that. The scene is yours.
Thank you. Thank you for the introduction. So, as he said, I'm Zoe Arrigoni, and I'm here to present you the result of my master internship. As a context, nowadays we know that there is a big plastic pollution. We can see on the photography. If we look now at the sources of this plastic pollution, we can see that there is a really big proportion that come from automotive field. But is this all lonely tire really responsible for this big proportion of pollution? Is the question that we can ask. Indeed, there is another sources that is less known of plastic pollution from tire. We will have the abrasion of the tire on the road surfaces that would generate little particle that we call tire road wear particle. And this particle has a chemical compounds, composition really diverse, as we can see, because those particles are a big mix of tire particles, of brake particles, and road wear particles. Those particles are sized between micrometers to millimeters. How does those particles end up in the environment? Those particles generated by the tire would be transported by the runoff water and end up in the aquatic environment. Those particles could sediment and be ingested by gammarids that are benthic invertebrates. They can also leak their compounds, and those compounds that are very diverse, as I just said, have all their own dynamics, so we make the situation really complex. Those compounds can be absorbed by the organic matter, but also be absorbed by the inorganic matter, and also be transferred directly to the gammarids or other benthic invertebrates. So the objective was to evaluate the toxicity of road dust and tire wear particles, and also their additives and the product of those additives. Why it's a concern? We know that the additives have neurotoxicity effects, so we have the hypothesis that it would impact the behavior. We know for fact that it has effect on the vertebrate, but what about the invertebrate and the gammarids that are directly in connection with the particles in the aquatic environment? To conduct these studies, we created microcosms that are a simplified ecosystem in the lab. It was only just sand, water, and the gammarids that you can see. And the little black dots are the tire particles. We separate male and female to have a differential between the sex. And we did a 21-day exposure to have a chronic aspect. We had three different conditions, so a condition control and a tire wear particle that was a standard that was given by the tire industry. And we had also road dust that we collected directly in the field to be representative of the environment. What we look at was different parameters. We look at the chemistry to evaluate the concentration in the water, concentration in the sand, and the concentration of the contaminant that we would find within the organism. Then we look at the ecotoxicology parameters, mostly mortality and reproduction. And now the thing that is really interesting us today, I will present you preliminary results about the behavior test. We did video tracking and feeding test. For the video tracking, I use Animaltea as a logical analysis. So for the results, this is supposed to be on the right, but whatever. We can see that we have a really big concentration of our contaminant in the sand and in the gammarid. So we can define some parameter. We can define a distribution coefficient. This KD said that there is more contaminant in the sand than in the water. And we can also define a bioaccumulation factor that says that there is more contaminant in the gammarid than in the water. So it's really a concern. Now, if we look at the ecotoxicological parameters, we didn't find significant results for the mortality or the reprotoxicity. About the feeding test was during a week, we would calculate the surface of the leaf. Then we will give them to the gammarids, and then we will collect them 1 week after to see the consumption rate after 1 week. And it was not significant. But now, more interestingly, if we look at the result of the video tracking, we can see that we have our two contaminated groups that are separated on the PCA from our control group. And if we look at the parameter, it says that the control are less active and also have more social interaction than the contaminated group. And also the tire wear particle was the highly concentrated group. It's also different from the control group on the social interaction. So it's means that we have really a significant result on behavior about our two different groups. So just to summarize, mortality and reprotoxicity, non-significant. It's not surprising because we worked at environmental concentrations. So usually ecotoxicological studies put a really high concentration of contaminant to have a really big effect, and we choose to be more representative of the what happened on the environment, in the environment. The feeding test was non-significant, but it's okay. Feeding tests are used daily by to monitorize the exits of water treatment plants. So it's a test really known, but there is another study about tire wear particles that also evaluate the feeding rate, and as in our study, they couldn't find an effect on the feeding rate. So our results are logical through the literature. But we went further because they didn't evaluate the behavior, and the behavior was a result significant in our study. So this is a really big, important things about ecotoxicology is we should go further in the evaluation of behavior, because the parameters that we evaluate usually are not sufficient to really understand what's happening with environmental concentration. And I found another study that was not on aquatic invertebrate, that was on terrestrial invertebrates that also show an effect on the a change on behavior, because the worms had an avoidance behavior through a contaminated sediment. So this is really encouraging. Well, it's not encouraging because we are talking about contamination and pollution. So we would like to have less significant results, but as a researcher, it's always interesting to see effects. So as a conclusion, those primary results have some limits, because I had a little few number of gametes. Also, it was only on the female, so I should check on the male to see if we have the same results on the PCA. And we focus on 3 contaminants, but as we saw, there are a lot of different compounds in those particles, so maybe we should check the other additives. And in a more global way, because gametes are bending invertebrates at the base of the trophic chain, maybe there is an effect of bioaccumulation effect through the trophic chain, so we should check that on. Also, because it modifies the behavior, maybe it will modify also the dynamic, the population dynamic. And as we saw with the worm studies, the aquatic environment is not the only ecosystem that is affected by tire particles. So maybe it would be interesting to link the transport of those particles to the terrestrial ecosystem. As a conclusion, I will say that I arrived in my internship with a few questions, and I end up with a lot more. So thank you for your attention. Do you have any question?
If I remember correctly, gametes during mate choice, the male, they kind of try to, there is homogamy on the molting time, because they need a female who's gonna molt, and just after molting, they can fecundate them. So I was wondering, like, if the pollutant has quite an impact on locomotion, maybe it completely changes also this aspect, like the mating dynamics, and whether they are capable of mating or not. And did you plan to look at this in the future, or?
Well, I think it would be really interesting to look at this particularly, because we wanted to see the effect on the reprotoxicity. So we contaminated just after this state, when the male and the female separate, as this, we could separate the male and the female in the microcosm. But it would be interesting to go, yeah, as you said, just before the mating state, and to see if there is a difference on the locomotion aspect. Thank you for the idea. I will look at that. Other questions?
Thank you for the talk. Really cool. I also study gamma rays for my master thesis a bit a long time ago now, but there are cool species I think to study in general. So I was wondering, because now basically you don't find any effect on the mortality of the gamma rays.
Yeah.
But you find this behavioral effect. So I was thinking maybe it would be interesting to see in a context of with predator how actually this mortality could fluctuate, because especially if it affects their behavior, you maybe it affects other behavior which might be risk-taking, things like that. And then at that moment you would actually see maybe an effect also on the mortality of the species. What do you think?
Yeah, I think it's a really good idea. I'm still new to behavior experiments, so I don't know how I will design this experiment. But I think it could be a really good idea. I wanted to evaluate the predator aspect by just disturbing the video in the middle of the video, like throwing a little rock or wood. But I didn't manage to do it on time. But I think it could be really interesting. I don't know if you have like any proposition to
We
share.
can talk later if you want about it.
Thank you.
Can see. Thanks.
Other questions?
Thanks for your talk. Okay, first thing first. This is the end of the behavioral session. So thanks again for our wonderful talks and all speakers and keynote speaker.